Tonight, however, we have a very distinguished panel who's going to be talking about gene editing in animals, and I'm going to turn over the rest of the time to my colleague Samantha Noll, who's a professor of biological ethics in the School of Politics, Philosophy, and Public Affairs. Samantha.
Thank you very much. Today we have a distinguished panel who will be discussing designing animals. Our first speaker is Dr. John Oatley. He's an associate professor in the School of Molecular Biosciences in the College of Veterinary Medicine here at Washington State University. He's also the director of Washington State University's Center for Reproductive Biology and the head of the Functional Genomics Initiative. He received his PhD from WSU and finished a postdoc at the University of Pennsylvania. And he's passionate about functional genomics and is currently doing several exciting research projects using CRISPR to help address global food security issues, and he has a robust research program. Now, according to ResearchGate, because as a millennial I have to Google stalk people, he has over 93 publications, including a recent write-up in Nature on a surrogate sires project. And you can actually pull that up and read that free of charge. So if you're interested, please check that out. Dr. Paul Thompson, as the W.K. Kellogg Chair in Agricultural, Food, and Community Ethics at Michigan State University. He received his PhD in philosophy from Stony Brook and held positions at Texas A&M and Purdue University. His research focuses on ethical and philosophical topics in food and agriculture, and he's the author of over 200 articles in refereed journals and at least 10 books. Many books. Dr. Aleta Quinn is an assistant professor of philosophy at the University of Idaho, so she's, hails from the Palouse. She earned her PhD in history and philosophy of science at the University of Pittsburgh and completed fellowships at the Smithsonian, Notre Dame, and Caltech before coming to the Palouse in 2017. She's a hobby herper, so she's inordinately fond of salamanders, so we can have that discussion. Okay? And her primary research is on the history and philosophy of biology, and in particular, phylogenetics. So let's give the speakers a round of applause. So what we'll be doing today is each of the speakers will be giving a brief presentation, and then we're going to open the floor up for questions and discussion.
Want me to go?
Yes.
All right. Well, I want to scoot behind you because I don't want to see. All right. Well, thanks, Samantha, for the introduction. It's rare that I get called distinguished, so I feel good. Hopefully that will carry through into what I'm going to talk about. So I'm a faculty member in the School of Molecular Biosciences. I'm trained as a germ cell biologist, and I have interest in germ cells because they provide this eternal cellular link between generations. All of us were germ cells at one point, right? And this is how we inherited genetics from our parents. So when we want to change the traits, the appearance, the behavior, the function of animal populations, we have to modify germ cells so that we can create animals that way. And we've been doing this, as I'll show you, for over 10,000 years. We just do it in different ways now. So I have a research program that's focused on both basic biology, but also in developing applicable tools to improve food animal production to help feed the growing world. And I'll show you some statistics of why we're interested in that. So I figured I'd start by talking about what genetic engineering is to hopefully lay the groundwork here. So we can define this quite broadly as being the direct manipulation of an organism's genome. That's the collection of all the DNA elements that encode for proteins, that carry out cellular functions, that make you live. And so if we have human intervention by way of manipulation, then we can consider it genetic engineering. How do humans influence the way genomes are put together in an animal? So we can tinker with DNA, but in order to take that DNA and create an animal off of it, we have to do reproductive processes. We have to get a sperm and an egg to meet. And and that way we can transmit genetic information. So we can really trace the dawn of animal genetic engineering all the way back to the domestication of animals. And archaeological evidence suggests that that first happened with the dog somewhere around 15 to 30 plus thousand years ago. And when we domesticated livestock, it was roughly 10 to 11,000 years ago. And livestock being food animals. First starting with sheep, and then with the goat, and then pigs and cattle. And so really the first time that we as the human race went into a field or other open space and said, I want this male and this female to breed together, so I'm going to confine them in an area because I think that the combination of their genetics is going to produce offspring that have something I want. Whether that's companionship or hunting in the form of dogs, or whether that's food products or use of animals for power. We took away natural selection and we decided to create a genome, a combination of genetics that may not have existed in nature by way of human intervention. That's the dawn of genetic engineering. And so we can really trace this as an evolution. We can trace back to selective breeding, which is what I just described, selectively having individual animals mate together so that we can produce offspring that have something we think we want. We can trace that back to 10,000 BC, and we did this for thousands and thousands of years, really up into the 1980s. And it's the 1980s that we really started to go after what we call genomic selection, looking into the DNA, looking into the genome of animals and selecting animals to breed based on that information. This led to the development of what we call transgenesis or transgenics. As I'll define here in just a minute, that involves recombinant DNA. The 1980s, early 1980s was the first time that animals were created with transgenes. And transgenes require recombinant DNA. And leading up into now, transgenics was the primary way that we created genetically engineered animals, all the way up until about 2013. And that's when gene editing burst onto the scene. And so these were tools that were adapted from microorganisms to be then applicable in mammalian cells, where we could go in and modify DNA in a more precise way without the need for recombinant DNA. And so I'll show you that in a little bit. And so that's where we're at right now. This really isn't that old of a field when we talk about gene editing of animals. Right? It's only about 6 going on 7 years now. And we've applied this evolution in all facets of animals, whether it's from rodents to create models that we can study in the lab to better understand biology or how diseases arise within animal populations or human populations. We apply this to companion animals such as dogs and cats and to livestock for improving food production. And so really tracing this back to 10,000 years ago, we can even come and see paintings in Egypt of murals. This is from 8,000 BC, showing human beings influencing the way that livestock are behaving. And so we can look at this even more modern based on the way that animals, and cattle in particular, have changed their appearance. So these are 2 animals that were the grand champions in 1835, which is the big animal here, and then the grand champion cattle at a state fair in 1937. You can see in that roughly 100-year period of time, we changed cattle from being really big and kind of fat to short and kind of fat, simply because of selection for a certain trait. Take that to the 1950s, we kept shrinking animals. You see this guy here? It doesn't really look like cattle, it looks like a furry pig because it's really short and fat. And then for some reason, 30 years later, we decided we wanted to be really big again. So we selected for larger frame size and engineered the genomes by selective breeding to be quite tall. And now we come to modern day, we've started to shrink them again, so now they're even shorter, and somehow we've selected for this weird sway in the back, right, which probably has a link to something more beneficial, like the meat tastes better. So, just by selective breeding, we've changed the phenotype of animals dramatically over roughly a 200-year period. So why do we do this? Well, by and large, we try to change livestock populations because of trying to address food security. So when you talk to people in North America, you don't hear about food security as being a major issue a lot of the time. We don't see it very much. And that is because in high-income countries like North America, over 2 billion people are classified as being overweight. That's 35% of the world's population. It's a lot of people that are overweight. 90% of those individuals live inside high-income countries like the United States. There's roughly 1 billion people in the world that can be classified as malnourished. That's about 15% of the world's population. 90% of those individuals live outside high-income countries. So outside countries like North America, food security is a big deal. In fact, more people every year die by starvation. This is deaths per year worldwide in the millions than cancer, malaria, foodborne illness, or HIV and AIDS combined. More people die by starvation than all of those other afflictions combined. And yet, the amount of dollars that are being spent on cancer research is 10 to 20 times more than on food security. But that's it. So why is this a growing problem? It really is a global food challenge, okay? And so what I've plotted here is the world population in billions of people from the year 1900 with a prediction out to 2050. Somewhere around the year 1940, the world's population began doubling every 40 years. So 1940 to 1980, we doubled the world's population. Next year, we will have doubled the world's population compared to 1980. And so for most of you in this room, 40 years from now, there's going to be twice as many people in here. You're going to have to find more chairs. Okay? So how do we feed people? We have to increase, and so let me back up. We will hit over 10 billion people on the planet by the year 2050. To feed that number of people, we have to increase our agricultural output, both plant and animal, by at least 60%. So one argument can be made, I'm just going to make more food. Probably is not going to work, and I'll tell you why. This is the percent of land that is currently being utilized for food production, whether that's agricultural grounds for producing crops or grazing lands for livestock. And this is the percent of land that is not in utilization. You can see in 1950, roughly 70% of land, arable land, was open for increasing agricultural output. Out to 2050, roughly 97% of all grazing lands will be in use. And approaching 90% of all cropland will be in use. So it's not a simple matter of just increasing production. There's not enough land available to do that. So this really is a global problem. We have decreasing agricultural land, but an increasing demand for food. And animal protein has been and will continue to be a major demand in the growing world population. This is the percent change in demand for animal protein from the year 1970 to 2000. It grew by 70%. Between 2000 and 2020, it will have grown another 50%. And by the year 2050, when we have 10 billion people, it's estimated that it will have grown another 50% from there. So the demand for animal protein in the form of meat or dairy products continues to rise and rise and rise. Arguments can be made that we should be transitioning to plant protein-based diets, and those are legitimate arguments. But the fact of the matter is, in developing worlds, animals get more bang for the buck. It's very hard in some non-arable regions of the world to produce plant protein. So, thinking about how do we increase the efficiency by which livestock produce products for human consumption. That's where we think the solution is. Not just producing more, but enhancing the efficiency by which they do that. So how do I define that? Livestock production efficiency is how well animals can take the inputs we put into them such as water, which is becoming more scarce all the time, disease prevention, like antibiotics, or feedstuffs, like grasses or crops or grains. We put these things into animals, and then we ask them to produce a measurable output for human consumption in the form of meat, milk, or fiber. The one intrinsic variable by which the animals can convert these inputs into these outputs are their genetics. This influence the efficiency by which they can take these inputs and give them into an output for human consumption. So the key really is to maximize the output with minimal input. We don't want to keep pumping them full of antibiotics. Drives antimicrobial resistance. We have less water to put into them. We have less quality and amount of feedstuff to put into them. So we have to maximize their output with minimal input and prevent the onset of progression of disease. Of course, animals are left asking, how do you expect us to achieve this? This is where human intervention comes into play. And so the human intervention principle is that we can influence genetic gain. That's the change in the performance of an animal from generation to generation that we produce by selective breeding or something like gene editing. This can enhance their ability to create outputs, and we can feed more people. We've primarily done this by selective breeding and propagating desirable genetics. We do this by widespread dissemination of gametes, sperm and eggs. So if we have an individual animal within a population that has the traits that we want, and we want to have that animal have an influence on a worldwide scale, we can collect sperm and eggs from them, ship them around the world, and say, use these for purposes. One of the places this has had a huge impact is with artificial insemination. And what that means is that I can take a bull that exists in North America, collect sperm from this bull, and put it in a frozen state, and I can ship it around the world. And it can be used to breed thousands of cows and propagate that genetics. This was commercialized first in the 1940s, and the one place it's had a huge impact is in dairy cattle production. In fact, roughly 80% of all dairy cattle around the world are bred by artificial insemination, not natural mating of a bull mating a cow. And so if we look at the pounds of milk produced per cow per day, in 1940, when artificial insemination came on the scene, and then last year, 2018, it's a quadrupling. The average dairy cow makes 4 times more milk now than she did in 1940, primarily because of selective breeding and propagating unique genetics. In fact, we have in North America a surplus of milk. We throw away almost more milk than we actually consume. Right? Why it doesn't go to other regions of the world, I have no idea. That's something I can't solve. So, artificial insemination and selective breeding is pretty good, but there are challenges. It's primarily based on something I can see, an observable phenotype. That animal grows efficiently, that animal has an udder that I think is going to produce lots of milk, or they have a temperament that I like. It's an observable phenotype. Doesn't always match up genetically. It can take 5 generations and 10 plus years to achieve an incremental change within a population. So if you think about the next 30 years, when we hit 10 billion people, it's going to be hard to make a major impact using this type of strategy. We can also get genetic drift or have inadvertent selection of negative phenotypes. One prime example of this is this bull right here. This is a bull that exists in North America, well, not anymore, but he did. His name was Rito 9J9. He was used to breed roughly 70% of beef cattle in North America. What wasn't known is that he carried a recessive mutation that led to this syndrome called curly calf syndrome, where these animals would be born with deformations in their limb formation. And they didn't survive very long. However, we selected this animal because we thought he made really good steaks. We didn't know that he carried a recessive mutation for this. And so this is where selectively, based on a phenotype, without screening of the DNA, we inadvertently selected for a bad phenotype. So this has led to what I would call second generation approaches, where we can screen for DNA variations in the genome, use that for rationally selecting livestock, but that's also met with several limitations. And it's because these variations don't always lead to the right phenotype we're looking for. So this has led to a third generation approach where we use molecular tools where we can come in and modify DNA to do efficient and precise genetic engineering. Conventional approaches to achieving this have involved recombinant DNA. Recombinant DNA is DNA that does not exist in nature without human intervention. This is where we take DNA from two different organisms, stick them together, and we make it recombinant. We recombine them. It's artificially produced DNA. And we can introduce that into an embryo. And if that DNA integrates into the DNA of the embryo, we can then produce an animal from this, and it becomes transgenic. We can also take this recombinant DNA, put it in a cell, in a culture dish, take the nucleus, or the genetic material out of that cell, and put it into an egg where we've taken the nucleus or DNA out of it. That's cloning. Both of these things have great promise, but they've had limited payout for several reasons. One is the social stigma of genetically engineered animals and recombinant DNA. If you Google genetically engineered, genetically modified, you can come up with things like this, where Monsanto is taking us from Neanderthals all the way to modern man and then back down into pig form, or strange anomalies that might form in animals, or cows with humanized teeth. This is some of the nicest veneers I've ever seen. What I really like is the cattle don't actually have teeth on the top. Like, they have a hard palate. So this would be amazing. And I think it's because recombinant or foreign DNA are actually scary words for a lot of people. Potentially rightfully so. But they don't understand what this means. And so when we say we're putting something foreign into the genome and creating an animal, it conjures up a lot of emotion and worry. There's also technical challenges, and that is that if we want to modify embryos, pretty easy if we're working with something like a mouse, but this is an embryo from a pig, very hard to figure out where to inject DNA. Cloning is very expensive, it's very inefficient, and oftentimes leads to birth defects. So this is where the game changer of gene editing came along, in the form of several different approaches. The most high profile is CRISPR-Cas. Gene editing is indeed recombinant DNA free. We can modify the genome without having to use recombinant DNA. And it's very precise. It's very simple and efficient. We can apply it to all animals. It involves two components. One is called a guide RNA. This tells the protein Cas9, which is a protein that breaks open DNA, where to go in the genome. And if we introduce this into a cell, it'll break DNA, use natural repair processes that happen every time DNA is broken. Every time you're exposed to UV light, you break DNA as well. And these natural repair processes are often air-prone, and they can bring about a genetic change. So if we introduce these CRISPR-Cas components into a single-cell embryo, this is a one-cell embryo, we can then convert that embryo into a gene-edited animal. And so CRISPR-Cas really is the tool in the toolbox that researchers have been searching for efficient and precise genetic engineering of food animals. The leading edge, when we talk about livestock, to address the future of food security, can roughly be put into four bins. The first is addressing animal welfare. You probably hear a lot about this with taking the horns off of cattle. Can we prevent that with gene editing? Disease resistance. Can we make animals resistant to pathogens so we no longer have to pump them full of antibiotics? Enhancing reproductive capacity. As I've already talked about, we pass genetics between individuals by reproductive processes. And then lastly, can we make them grow more efficiently so we can put less inputs and get the same or a greater measurable output? And hopefully this addresses food security. I'm just going to tell you about 3 examples really fast. The first is porcine reproduction and respiratory syndrome, or PRRS, resistant pigs. PRRS is a syndrome that pops up every so often throughout the world's pork populations, and oftentimes it results in the elimination of anywhere from 30 to 60% of the world's pig supply. This impacts how well we can feed people. A group at the University of Missouri found that the virus that causes this syndrome interacts with this molecule on the surface of cells in the pig and they used CRISPR-Cas and knocked it out. And what they showed, this is not all that complex, is that pigs that were not knockout, what we call wild type, this is the incidence by which they got sick from the virus. Most of them did get sick, but when they knocked it out with CRISPR-Cas or made it so it didn't function anymore, none of the pigs became sick to PRRS. So we can put pigs into the human food supply right now that would be resistant to this virus and stop having these outbreaks around the world. But we can't do it because the federal policy is not in place to allow these animals into the food chain. From an animal welfare standpoint, if you go to a dairy operation, you probably don't see many animals like this that have horns. And that's because more than 90% of dairy cattle have their horns removed when they're younger. A painful process causes distress and sometimes disease. A group at a company called Recombinetics figured out how to use a different gene editing process to take out the gene that's needed for horn development. So these horns don't need to be removed anymore by mechanical or surgical processes. Again, these animals are ready to go into the food chain, but they cannot be removed. From a reproductive biology standpoint, reproductive capacity at Washington State University, we're working on a project called Surrogate Sires. This is where we're trying to improve the capacity of individual males within populations to influence genetic change on a big worldwide scale. I'm just going to show you one slide to depict this. What we try to produce in places like North America are cattle that look like this. We call them black baldies because they have the black coat color but this bald face. These are produced by combination, by selective breeding of a black Angus animal and a Hereford. They have great qualities for meat production. The problem is trying to get this one male to have an influence on large populations. One of the ways that we can do this is by sperm stem cell transplantation. So we can transfer the germline that makes the sperm of this individual into a bunch of surrogate recipients that can then breed by natural mating to produce these offspring. So the keys here are that we have to make these surrogates that lack their own sperm production. And we've done this with several species now by way of genetic engineering. These are the ways we do it in mice. They don't really work, and so we try to do this with genetics by using CRISPR-Cas to knock out key genes that are needed for the surrogate to make their own sperm, and if we knock them out, then they can't. And we've done this at Washington State University now with several different animal livestock species, including pigs. This is a CRISPRed pig. He's totally normal size, does everything that a male pig normally does, but his testicles don't actually make sperm. These are some calves that we had born just a few weeks ago that are also gene edited with the same gene as this pig. One boy and one girl, totally normal, growing fine. Here's some goats that we also use CRISPR-Cas to knock out the same genes. And so none of these males produce their own sperm, but we can transfer another male's sperm so that we can influence genetic gain within populations by breeding. And so in the end, we think that gene editing really is the future of food security as we continue to grow the world's population. We believe we can use it to create designer livestock so that we can have minimal input with maximal output, matching the animals to their environment that we ask them to thrive in, and make them resistant to pathogens so that we can hopefully provide cheap, nutritious, and safe food for this 10 billion plus people we're going to have in the next 30 years. There are challenges ahead. The first one is technological refinements. Every year, I told you gene editing is only about 6 to 7 years old. We are improving the efficiency and precision almost every month. There's a social acceptance aspect of this. We believe we need a new narrative to talk about gene editing. We can't lump it in the same bin as transgenesis with recombinant DNA. We need to have education about what gene editing actually is, and we need to take into consideration the ethical aspects of this. I was at a summit a few weeks ago with both Dr. Noll and Dr. Thompson. And Dr. Thompson said in his talk, I really wish basic researchers would take a moment and thank the animals that gave their lives for the research. I'm going to do that. I'm going to take a moment and thank all of the animals that we've had to sacrifice to get to where we are now. I do take that into consideration. And the other aspect is the government policy. Right now, the infrastructure doesn't exist at the federal level to allow gene-edited animals into the food chain. We've been working with the USDA and FDA in a collaborative and transparent way to try to disseminate or transfer the knowledge that we gain in the research process to the federal regulatory agencies so that they can develop a structure that will allow these things into the food chain. So I'm going to leave you with this quote because I love it. And it is, "I know of no pursuit in which more real and important services can be rendered to any country than by improving its agriculture, its breed of useful animals, and other branches of husbandman's care." This was in a letter that George Washington wrote to the Continental Congress in July of 1794. I think it was important then, and it's important now. And this was almost the genesis of land-grant universities like Washington State University, to try to improve agriculture production within the United States. Thank you.
Thank you very much. And next, Dr. Paul Thompson will be talking about and elaborating on some of the ethical considerations concerning biotechnology design of animals.
Well, I am going to stand up and use the podium because then everybody can actually see me. But I don't have slides. I am going to concentrate on some of the policy issues and some of the ethics issues that are associated with gene technologies and specifically gene editing. And I want to start by talking a little bit about the policy. And this really is kind of an explanation of why we're here today, right? You know, when Dr. Oatley talks about not being able to get these animals in, I want to kind of explain a little bit more about what that means and why that is the case. The policy really goes back all the way to the Reagan administration. That's when biotech first started to be discussed. And during the Reagan administration, there was a policy decision made that we would not pass new legislation or create new regulatory agencies for products of biotechnology. We would use the existing suite of laws that we use to regulate all new technologies. And this includes the Toxic Substances Control Act, which actually turns out not to be very important in biotech, but includes the EPA regulates pesticides under a statute that we all refer to as FIFRA. I'm not even going to spell out that one. And the FDA regulates under the Federal Food and Drug Act, and the USDA regulates under both some food safety, meat safety rules, plus also some the Plant Protection Act. So these acts had to be sort of reinterpreted to be applied to biotechnology. During the context of this, we actually have had fairly successful experience with plant biotech, and we have a lot of plant biotech products that are on the market. We've all been eating them for probably for most of the people in the room all your lives. I've been eating them for only about half my life, but that just reflects the age difference here. So, we've had very, very few animal products on. I'll try to explain why that is the case. However, with the advent of the gene editing technology that has been just been described, many of these agencies are revising some of their regulatory requirements because this technology is thought to be much more precise and much less prone to introduce unwanted side effects as a result of the gene editing technology. And so some of the ways in which agencies are regulating plants have already basically cleared a number of gene editing of plants to go through the regulatory process much more quickly. However, there was right at the outset an issue with animals, because it was not at all clear how we were even going to review animals that had had their genes modified. It was difficult to see what statutes would apply to them. And there were kind of two general areas in which this was being discussed, one of which was animals that are used as models in research. And there's been a lot of application of animal biotechnology to mouse models and animals in research. This turned out to be less problematic because the National Institutes of Health and the U.S. Department of Agriculture do have some regulatory oversight over research animals through the use of animal care and use committees. You've got one here at Washington State. All institutions have one, and so for research animals, that aspect of the oversight was adapted for that purpose. But for animals that are going to be sold, whether for food or for whatever else you might want to sell them for, some animals have been modified to actually produce valuable substances in their blood or milk, and those have actually gone through. There was not really a real clear regulatory pathway for that. And ultimately, after a lot of debate, the Food and Drug Administration decided that they would regulate animal genetic modifications of animals as under their authority to regulate new drugs for animals. FDA regulates new drugs for humans. They also regulate new drugs, veterinary drugs, drugs that are used for animals. And they basically made this decision because there's some language in the statute that basically gives them the authority to oversee anything that modifies the structure and function of the animal, and the genes do that. Right? Now, the problem with that, from the standpoint of some of the applications that Dr. Oatley's been talking about, is that getting an approval through the animal drug process is time-consuming, requires a lot of data, and it makes it extremely expensive. Really, the only food animal that has managed to make it all the way through this are the fast-growing salmon. Gosh, how long did that take? It was like 15 years. Now, it probably wouldn't take 15 years to get an animal through that, but it would take a long time. It would require a lot of submissions. So there is a regulatory pathway that you could get these animals onto the market, but it would require generating a tremendous amount of data that would then be very carefully reviewed at FDA. It would require clinical trials and a lot of stuff, and so it would be very expensive. It clearly would discourage applications that didn't have a pretty significant economic benefit tied to them, and it would be certainly beyond the reach of any university to cover the cost of moving an animal through that regulatory environment. So there has been kind of a push, and this summit that we were at a few weeks ago in Washington was really exploring some ideas for ways in which the coordinated framework could be modified, and frankly, it may take new legislation. We all know how easy it is to get legislation through Congress, right? That should be just a snap, right? It may actually take some legislative change that would actually create some alternative to the animal drug framework. So there, as Dr. Oatley mentioned, there's also been quite a bit of ethical concern about genetic changes in animals. I've been at this a long time, and I was actually at a meeting that was held in 2001 on animal biotech prior to FDA's decision about how they were going to regulate these animals, was sort of tasked with giving the ethics talk there. And the talk that I gave, let me actually go ahead and tell you this whole story. So I was at this meeting, gave this talk. I'll tell you a little bit what I said in a minute, but I got back, and like 4 or 5 days later, I wake up and I'm listening to National Public Radio's morning edition on the radio, and I hear the reporter talking about genetic transformation of animals, and it sounds like he's interviewing some of the guys that were at this conference with me. And so I, turn up the radio and listen more carefully, and next thing I know, he's interviewing me, and basically what he had done is he'd taken a recording of the talk that I gave and sort of made it, cut it so that it was like an interview. And what I said on the radio was, I'm not sure I'm going to get this exactly right, but I had I had, and he set up my piece by saying that there's a special issue about genetically engineering animals, which is that animals have feelings. And I in my talk, I had talked about some breeding work that had been done with hens, with chickens, and there was it was actually done at Michigan State back in the 60s, I think it was the 60s. They created a strain of congenitally blind hens. The hens breed true, and they're blind, they just can't see. And so there had been some discussion that we should be using blind chickens in egg production because the blind chickens couldn't see each other and wouldn't mind being crowded together. And then I said, but everybody thinks this is ethically an ethically horrible thing to do, right? That was the end of my thing. That was my 15 seconds of fame on the radio. But if you ever do get quoted on Morning Edition, I can assure you that you will hear from friends that you haven't talked to in decades, those of you who are more than decades old. And I heard from them. I also got criticized by animal activists who said that I had advocated blinding chickens on the radio. And trying to start a campaign against National Public Radio for even allowing this crazy professor who is advocating blinding chickens. And I got complaints from the National Chicken Council, which is the trade organization for meat birds. At any rate, part of my point was that even if it turned, actually blind chickens are not a good idea, they aren't better off. But even if it had been, there was just something weird about that change that really rubbed people the wrong way. And that was really just try to give a sense of how certain kinds of ethical issues would be associated with genetic changes. Now, I'd say the hard work on this was done by another philosopher named Bernard Rolland, who, Bernie's still with us. He's on both the veterinary faculty and the philosophy faculty at Colorado State University. And Bernie had started writing on genetic engineering of animals back in the early 1980s. Probably his most important work is a book called The Frankenstein Syndrome, which goes back to 1995. And Bernie had proposed what he calls the principle of welfare conservation. So Bernie's doesn't think that it's always wrong to genetically modify animals. What the principle of welfare conservation states is that when you transform an animal through genetic modification, the animal shouldn't have worse welfare than another animal of the species, right? So you shouldn't be using this technology to produce animals that suffer more. The example of the photograph that was shown would have been an example, and you can do this through ordinary breeding. There were a number of examples in the 90s of experimental uses of gene transfer that did produce very dysfunctional animals that had to be euthanized, and so this was kind of a front-of-the-mind concern for people. And I would say there was a fairly widespread agreement that this was, in fact, the right way to think about gene technologies, that they could be acceptable, but the ethical test was that the animals had to be at least as well off as other animals of the same breed or species. Now, it's probably important to note that you can produce dysfunctional animals through conventional breeding. And there is no oversight or nothing that would prevent people from doing that. And we have done that. We have produced, going back to the broiler chickens, we had chickens that had tremendously weak bone strength and suffered from all kinds of injuries. The breeding companies have, I think, now addressed some of those issues, but you can produce a dysfunctional animal through breeding. So I'm going to sort of come to the idea that one of the ethical issues that we face now is we sort of have to make a choice between looking at gene technologies according to what Rolland recommended. In other words, we're going to apply these technologies, but we want to make sure that we're actually promoting animal welfare with them. But that actually, in a certain sense, it is not entirely fair to gene technologies, because we're allowing other technologies that do produce dysfunctional animals. And so in a certain sense, we have to choose between an ethical principle, which is really focused on the welfare of the animal, and an ethical principle that's focused on being fair to technological innovators, right? Should we subject people who are using gene technology to animal welfare tests that we don't apply to people that are using selective breeding? And I think in the U.S. that's how we've tended to look at it. We've tended to say, let's not apply a standard to one technical means that we're not applying to another technical means. Whereas I think in Europe, and to some extent throughout the rest of the world, they've tended to go the other way, and they're just not willing to allow new things. They say, yeah, these bad things can happen, but we're not going to allow some new way that animals wind up being worse off. And I think that's still an unresolved issue from the ethics side. I also want to say a little bit about, one other thing is that, in Bernie Rollins' version of this, this principle of welfare conservation was understood as what philosophers call a prima facie principle. That just means that, yes, this is where we start our consideration, but the principle could be overridden by considerations like in medical research, where you're working on a disease that may be having very significant health effects for humans, and the benefits to humans from developing a new therapy or a new drug would outweigh the issues for animals. And perhaps we could have similar kinds of arguments with some of the feed the world kinds of arguments here. So the idea there is that what this principle does is it creates a burden of proof that has to be overridden by an argument that there's some sort of compelling benefit that would allow us to not meet this rule. I want to talk just a little bit about the reaction to some of Rollins' work, and primarily outside the United States, primarily European philosophers, but a number of philosophers and animal activists were didn't feel like Rollins had gone nearly far enough, and they felt like there's something wrong. Maybe they're sort of stirred on by my blind chicken example or something like that, but they were kind of searching for some sort of alternative rationale that would provide much stronger prescriptions against genetic engineering. And this is a pretty complicated literature, but I think that I can summarize it by saying that they sort of coalesced on this idea of animal integrity. That what you're doing when you're genetically modifying an animal, perhaps even in a way that's consistent with the Rollins welfare conservation principle, is you're violating the integrity of the animal. And some of the arguments for this, frankly, just didn't make a lot of sense to me, but I'll try to kind of give you the most charitable interpretation of it I can. And I think that basically what people who are proposing this idea of integrity were calling attention to is that there are relational aspects to our ethics. This idea that ethics has relational aspects goes all the way back to Aristotle. We have ethical obligations to our family members, and we have ethical obligations to our friends, and we have ethical obligations to our countrymen. And perhaps we should be a little suspicious of standards like things that focus only on welfare that seem to be sort of universal and don't recognize some of these specific relational considerations. So the way that this would apply with respect to animals, a lot of our relations come from the history that we have with others, whether they're people that we know personally or people that we share a common history with, in the sense that we're all countrymen or something like that. They were arguing that we have a special history of relationships with animals that's been characterized with our domesticated animals, our farm animals. It's been characterized by a certain reciprocity. In other words, the animals are getting something out of this deal as well as the humans. Even if the animal is ultimately slaughtered, domestication is something that several species of animals seem to have benefited from. The humans are taking care of them, they're protecting them from predators, and to go into more Native American types of rationales. The Native Americans, I heard this story just the other day, will talk about the animals that are hunted as giving themselves to the people, and that there's a kind of exchange relationship that's going on. Perhaps the history of breeding is in some respects an exchange relationship that reflects a history in which the animals have a certain amount of agency or activity in this. And the concern was that with gene technologies, we see that slipping away. I would also characterize this as a trend towards a more complete instrumentalization of the animal, rather than seeing the animal as a living being, as something that has consciousness, capable of feeling pain and a certain sense of its own life. It comes to be viewed just as a means to an end, just as a means to getting something to eat or some other sort of product about it. So I think pulling together these notions of relationship and that animals are becoming more and more like mere tools, we can make a little bit of sense of this notion of animal integrity. I'm not sure I want to completely defend it, but I do think that it's calling something about our relationship or the way that we might or should relate to animals into question. And so that was partly in the spirit of the suggestion I made at this summit, that there are probably some things that we could do that would sort of remind us of the relationships with our animals that would not actually be that costly. I pointed out that in the Christian tradition, before you eat a meal, you would say a prayer or ask a grace, and part of the rationale for that is that you're sort of expressing your relationship with the food that you're about to eat. And it's also a relationship that's a relationship with God, and you're acknowledging that your own life and dependence relies on the consumption of this particular animal, and that one of the ways that we might sort of block full instrumentalization of animals is to do something, not necessarily religious. I actually would discourage a strongly religious, but just something more like a moment of silence occasionally that would sort of remind us of this sense of relationship with the animals and having a sense of what would go on. I would also say that, sort of now turning to where we need to go, that we do have some big ethical issues that we need to wrestle with. In one sense, I think we need, philosophers love to talk about a national conversation, right? We need a national how the hell we have a national conversation? Pretty difficult. But the idea is that we do need to really sort out this sense. Do we really want a sense of level playing field for innovators to be the dominant norm that we apply when we evaluate technologies? Or should, what I would call more substantive types of goals, in this case animal health and welfare, be more selectively applied? In order to make that the dominant goal, we would wind up with making a lot of changes, and I'm sure Congress isn't going to make any of those kinds of changes. But it might make sense to selectively try to apply a norm that is more focused on animal welfare in some of these new technologies that are coming along. And I would not single out only gene editing, but a lot of technologies that are currently used in agriculture. In lieu of something like a national conversation, we have some institutions where you can do this, and universities are one of the main examples of this. The National Institutes of Health has really incorporated a very strong ethics program into all of its genetic programming, and it's difficult to get a grant from the NIH without some sort of ethics component on the grant. I mean, this is really only applies in the Genetics Institute. It's not NIH-wide. So, as you're doing this kind of work, you have an ethics component in which you would have programs much like we're having today, where you have these kinds of discussions going on, and you invite faculty from across the campus to really sort of, try out this idea. Is this idea of animal integrity, is that just loony? Does that make any sense at all? Right? I mean, you sort of sort that out in an actually structured sort of process. We have also, in the past, had presidential bioethics commissions. They've never looked at animal issues. They've only looked at human medical issues. The current administration doesn't have one, but for the last 5 or 6 administrations, there's been this national body that sort of discusses these things and then issues reports that sort of lay out the pros and cons and how some of these issues take place. And I would argue that these are probably things that really need to accompany some of the types of technologies that Dr. Oatley has described. So that's my remarks, and I think I'll sit down and turn it over to.
Last but not least, we have Dr. Quinn, who will be giving a presentation on her work on, is it artificial organisms?
Yeah, it's not even my work, it's just some cool stuff.
Awesome.
Yeah, so my work is on phylogenetics, which is just what species are there and how are they related to each other. This is more of a teaching interest, but as we'll see, unless I'm going to talk about places where new technologies put pressure on what even is it to be a species. And I mean, not that there's not already problems with defining species, but anyway, I want to thank you in particular for your remarks about the integrity of the organism, because I've come across that language and I could not make sense of it. So I really appreciated that.
I may be making too much sense.
Also, so my advisor was an Aristotle guy, so it helps me to okay, put your Aristotle hat on, and maybe you can make sense of this. Yeah, so here's an outline. I'm going to tell you about de-extinction. I'll tell you about a laboratory species, and then I'm not going to tell you the appendices. That's just there in case people have technical questions that science stuff can, help explain what a gene is, stuff like that. But I don't intend to go through that. Instead, here's de-extinction. So this is somatic cell nuclear transfer, which our first speaker spoke about. And so basically what you do is you take a cell from one donor and then an egg cell from a different donor. You take the nuclear material, specifically the DNA in the nucleus, and put that into the cell from the other donor. And then magic science stuff, literally involving zapping with electricity at one point. So it is kind of Frankensteinian. If all goes well, then you end up with a cloned organism. You also need the surrogate mother. They're working on an artificial uterus, but we're not there yet. So yeah, this is the procedure done with Dolly the sheep. As mentioned, there's a lot of messiness in there. This is a very clean diagram, but in fact you end up with many dozens of organisms that are not viable. And so that's one ethical issue already from the start. But if all goes well, you can use this for several different purposes. There's only 2 northern white rhinoceroses left in the world, and they both happen to be female. So if you could use this to clone that subspecies back, there's other ways of doing that, but anyways, that's one example. If you want to get science fiction-y here, I love Robert Heinlein. In his novels, every human person has a clone, one or more clones that is produced, grown, but never made conscious. And so the claim is it's perfectly ethically fine because there's no sense of consciousness there, meaning there's no person there, but there's a body that has all the body parts. And if I need a new liver because I like beer too much or whatever, you can take that from your clone. And because it's genetically almost identical, there's almost no chance that it'll be rejected. So that's very science fiction-y, but that's in the future, potentially going to happen. But for my purposes, you can use this procedure even if the donor of the somatic cells is extinct. and we can take DNA from dead creatures, and you can do this. And so if this species or subspecies is extinct, but you've got a close enough living analog, you can produce, yes, it's basically Jurassic Park, okay? But
Is there a lysine contention? No.
so how far away do we think we are from this? 40 years. It's actually already been done. There was a subspecies called the Pyrenean ibex that went extinct, and it was actually cloned back to life. So it was de-extincted. The animal lived in the lab for a couple hours and then died, so it was re-extincted. So that's, I mean, as a philosopher of science, this is like, to me, like, what is it to be a species? Is this the same species? Is this a different species? Is this, usually most of the definitions of species rely on having a coherent historical connectivity. So this is like a hiccup in that. And so that's sort of philosophically interesting. It's also ethically interesting, okay? For reasons that have been touched on. Like, this is a pretty severe intervention onto the integrity of not just one organism, but an evolutionary lineage. Okay, yeah, so the Australians want to do this with the Tasmanian tiger, species of cultural significance. There have been reports in the news in the last few days that people claim that they've seen living organisms of the Tasmanian tiger, although the last individual was thought to have died in 1936. But basically every few years there's a flurry of supposed sightings of this thing. But they are working to clone it back. Okay, the other example. Here's one example of a species made in a lab, maybe, depending how you want to use definitions. What you do, take a sequence of, this is a bacterium. It's got a pretty small genome. You can figure out what every single one of those base pairs is. It's a couple 100,000 base pairs, so you can get pretty good certainty. They actually assembled a copy of that genome in the laboratory from non-living chemicals, just carbons, hydrogens, phosphorus, all that stuff. They took out the DNA from a different cell, went ahead and put their manufactured genome into that cell, and the thing has reproduced hundreds of thousands, millions of times, very short generation time in these bacteria. Okay, so, like, I'm not sure what to make of that. Is this a proper species like any other, or again, this is a pretty severe intervention? But they went even further. So they started out with a genome. They started knocking out regions and see if you knock this out, does it still produce a viable organism that can reproduce and so forth. And they just kept doing a trial and error for the whole genome until they knocked out, and they got down to this minimum. It's got 473 genes. They know some of it's to do with the membrane structure and function, metabolism, and then there's some they don't know why, but if you knock these out, the thing doesn't live. So they got it down to this very small genome. They added some stuff, basically because they wanted to, and then, more on that next slide, but then, so they assembled that genome from non-living chemicals, and then they put that into the cytoplasm of a cell from which the DNA had been removed. So they still have to use originally living material to get this thing online. They're working to replace that part too, at which point they'll say this is a fully artificial life thing. But it lived and has reproduced, and they declared it a new species, which again is a little curious. For me, my work is mostly with animals and some plants, and I know how the species naming procedures work for that, but bacteria is like a whole nother level of complexity. So I'm not sure how the Committee on Bacteriological Nomenclature has responded to this. Here's what they added. Because you're working with genes, but the genes, okay, so ACGT, but those code for protein codes, you've got 23, some number of letters to work with. They had enough letters that they put the names of the researchers into the genome. They put a Robert Oppenheimer quote, he's the father of the atomic bomb, James Joyce quote, and a Richard Feynman quote, which they actually got wrong. So this is Feynman's blackboard when he died. He was at Caltech. I was at Caltech when I was reading on this, so I had to tell my students that, like, if you're going to do all this, just come on, get it right. And then some kind of Easter egg. It's like a website or something where if you find it and decode it, you can go there. And I don't know what that is. I don't know if anyone's found it.
Extra credit assignment.
Yeah. Okay, so I've been super flippant in describing this, right? And like, we're talking about generating life. We're talking about changing what a species even is. And there's been no ethics statement at any point in my talk, and at any point as far as I know, and like, but these are bacteria. Nobody cares about bacteria, right? I mean, they're small, they don't have feelings, they don't think. So the question becomes like, these technologies, well, okay, let's start with just what are we gonna use this for? So this laboratory, it's in Bethesda, Maryland, they claim one thing they're trying to get is they're gonna try to put genes in there so that these things will sequester carbon to fight climate change. That sounds great. Of course, your worry is if what's the off button, because if they want to sequester carbon, we're mostly made of carbon, and are they going to turn us all into these little bacteria, right? So, yeah, so there's going to be potential side effects, and not just ones that you can predict, but also unknown unknowns, where like what you call deep risk, where not only do you not know that whether the risk will come about, you don't even know what roughly percentage that risk is going to come about, and you don't even know all the forms of risk that you don't know that about. Okay, so this is potentially a huge deal. For me, like, okay, is that an artificial species? If not, what would make an artificial species? At one point, you know, does the source and nature of the chemicals used matter for whether this is life? And what does it take to be a species? I think that's, I'll just end with this one though, because in the spirit of like, this is a continuity, this is really radical, cutting edge, but it goes back, you know, we generated bananas 10,000 years ago, we generated corn many thousands of years ago, those are artificial species. American bison, basically every single buffalo American bison alive today has cattle DNA in it because they've gone through a genetic bottleneck many years ago. There's people who claim there's one little herd up in some mountain that hasn't, but if you go to Yellowstone, you see the iconic image of the Wild West. It's actually part cow, part domestic. They all descend from domestic animals. California condors, very successful species recovery project. Every single California condor is periodically trapped and dialysized because otherwise lead would build up and kill the bird. So every single condor has a number and is periodically trapped and run through a dialysis machine. Does that make it less wild? Does that make you value it less? So, you know, going down and the continuum, like the de-extincted species, do you value it less? The other form of argument that often gets made is that with the ability to de-extinct a species will come arrogance, like partly like just let everything go extinct because we could bring it back if we wanted to. But even apart from that, you know, that Tasmanian tiger, if we bring it back, every time you look at it, it's a reminder that, like, you know, we think of ourselves as in control of nature in that way. So in a more radical way than previously. So that kind of argument gets made. Not sure how I feel about that, but it's on the table. Yeah, and I think that's really all I have to say because I want to get to the panel discussion. So, thanks.
Yeah, so we have about 15 minutes.
Yeah, about 15, 20 minutes, yeah.
We have about 15 minutes for discussion. So I'm going to open up the floor. So if you're interested in asking any of the panelists a question, please raise your hand and we'll. Trish in the back.
Hi, thanks. I just had a question for John. Dr. Oatley.
I can see you.
I just wanted to ask you, there's been all this literature lately about the contribution of vegan meats to greenhouse gas creation, including CO2, but also methane. So if you want to, you know, feed people whose crops are actually failing now, because for the first time in the last, since 2015, there's been a record of increasing food insecurity now, because for so long it was going down, but the FAO reports now that it's gone up. by 25 million between 2015 and 2019. So if eating meat is actually contributing in a pretty big way to climate change, which is contributing to a decimation of agriculture in some countries, particularly countries in Africa, for example, then it might seem that actually promoting strategies for better, more efficient, etc., meat production for people to eat might actually not be addressing the food problem that you're suggesting, but contributing to it by exacerbating climate impacts.
Yeah, so two things. One is, by gene editing, one of the potential applications is to make animals produce less methane. So in that respect, you could impact that. The other is that I believe that the statistics on cattle contribution to greenhouse gas effects is skewed. And that is because 100 years ago, we had about 60 million bison, all producing methane, roaming across North America. They don't exist anymore. That's more bison producing methane than we have cattle in North America producing methane. So it's not necessarily the introduction of cattle production that has increased the contribution of methane to the environment. It's the addition of all the other things, right? So I would make an argument that cattle production doesn't actually impact it as much as we think it does.
So something like keep eating meat, but keep the oil in the ground, or something like that.
Something like that, but I mean, even crop production, when we're harvesting crops, we're producing lots of carbon emissions, right? Because we're burning lots of fuels. Every time you see one of those combines going across the field on the pollutes, you're contributing to environmental impacts on the environment. So I think it's a combination of things to combat that. It's not just stop eating meat, right? In other regions of the world, animal products are probably the main source of protein. And it's simply because small shareholder farms, like in sub-Saharan Africa or India, can have 3 or 4 animals and feed their entire family throughout a whole year. They don't have the resources in the ground to be able to produce crops for plant protein. So, that's where outside of North America, outside the United States, I'm not sure. I just don't know whether we can plan to transition those regions over to plant-based diets. Just simply because we don't get as much output per available unit of land, right? So I think it's a combination of both. Should we be finding a nice middle ground? I think so. And we shouldn't just be, I hope I didn't come across as saying we should just all be going to plant or animal-based products. No, I think it's a combination of both. But I don't think that we can get rid of animal production for meat or dairy products.
Thank you.
Dr. Oatley, I'm glad that you mentioned the American bison, and I'd like to touch on that for a second and sort of roll into something that Dr. Thompson brought up, which is the sort of double standard that exists between the regulations on gene editing versus the regulations for regular animal husbandry and selective breeding. So you mentioned that when the American buffalo was on the prairie, they were sort of a negligible impact on climate change. Their methane emissions weren't an issue. And we had more bison than we now have cattle. So then when you read through some of the environmental ethics literature, like from Leopold, Paul Taylor, even Dr. Valerius Geist, who's more an outside the box thinker in that arena, they sort of want to separate human ethics from environmental ethics based on this presupposition that captive cervids or domestic livestock is a thing that's happened and that it's good and that's sort of taken for granted. Why do you suppose it is that there is that double standard, and why haven't we, in trying to pitch gene editing, why haven't we sort of pitched the idea that selective breeding be held to the more stringent standards as opposed to loosening standards on gene editing? I'm sorry if that was a lot of questions at once.
Yeah, all right, let me see if I can figure out where to go with this. So, if I understand you correctly, you're asking why aren't we bringing more regulation onto selective breeding?
Right.
To kind of align with the regulation that we have on gene editing.
Right, in light of the fact that there are also like some big existential risks that have cropped up in the captive cervid industry, like we have mad cow, which turned into Creutzfeldt-Jakobs, and we have all these different types of scrapies and things like that. So it's not that the captive livestock industry is without suffering and issues and things like that, and certainly with selective breeding. So why are we not taking that into consideration when we talk about this double standard and sort of pushing back on the captive industry, especially intensive agriculture?
Yeah, I'm actually not a proponent for intensive agriculture. I think we probably need to go in the opposite direction of factory farming. But I mean, there's a reason we've gone to factory farming. There's a reason we haven't regulated selective breeding. I gave examples of artificial insemination, and that's essentially artificially changing natural breeding processes, right? There's zero regulation, right? We can go and artificially breed animals any day of the week without any oversight whatsoever, and introduce what I showed with curly calf syndrome, inadvertent genetic consequences. So should there be some regulation of those type of strategies for breeding for intensive factory-based farming? Yeah, probably. But should they be as stringent as what we are putting on transgenesis or putting on gene editing technologies? I don't think so, because a lot of those issues that can happen by selective breeding are also issues that would arise if we just let nature do its course and let animals just breed, right? And with gene editing, a lot of the mutations that are being created with gene editing are mutations that could arise in nature. We just may not have selected for them. The PRRS-resistant pigs, for example. Individuals within a population, a large population of pigs, could acquire a mutation that gives them a survival advantage to the virus. We just wouldn't know that without screening millions of pigs around the world to find that one rare individual. And then that one rare individual, how do you propagate those genetics throughout millions of pigs around the world? It's technically challenging. So I guess all to say is that those mutations could arise in nature. We just don't screen for them and select them. So if we put more stringent criteria on selective breeding and try to align it with gene editing, I think we're going to narrow our ability to really progress and improve production characteristics. Because if we want to get away from intensive factory farming, we need to make animals that can be more efficient in producing the products we want. We go to factory farming because we don't have to manage them as extensively. So it costs us less, right? And so we try to confine it so that we can feed them all a whole bunch of crappy food all at one time. But if we can, and put a whole bunch of water in them at one time, put a bunch of antibiotics in them at one time, if they're all out on a range, we can't do that. But if we stick them in a confined area, we can. So if we can make those animals grow and produce products more efficiently in a range type of condition, because honestly, if we want to open up those areas around the world that currently aren't farmable, we got to be able to put animals on those areas where they're not going to get very good quality feedstuffs.
Right.
So we have
Like
to,
the buffalo.
right, so we have to do it either by selective breeding or by gene editing to make them more efficient, if that's the direction
I see.
that we want to be going. That's a long-winded response.
No, I
I
like
hope
that.
I
That's good.
addressed all
Yes.
I could just add briefly that there are alternatives to government regulation, and part of the way that we've gotten out of some of the bone breakage issues and broilers is that it's been a cooperative agreement. The broiler industry is just a few companies. It's a very concentrated industry. So those companies, together with the breeding companies, basically just acted to resolve this problem. I wouldn't say it's entirely resolved, but it's made considerable improvements. And that's a kind of soft governance. It really didn't require government, and actually you do see among producer organizations some willingness to go there, right? The problem with producers is they want a level playing field. They want to make sure that if they're doing something, everybody else is doing it too.
Mm-hmm.
You can achieve that through government regulation, but in some respects you can achieve it much more efficiently just through cooperative agreements among producers. So I think there are ways to go that don't really require congressional action, that can actually be more effective than that. But the other thing I was going to note is that part of the answer to your question is it's not an ethics answer, it's a politics answer. I mean, there were people arguing that we didn't need any regulation of genetic engineering. So a guy at the Hoover Institute named Henry Miller, who worked for FDA for a while, who has made that argument pretty vociferously. And if Henry were here, he'd tell you that the reason we have a regulation is because the big biotech companies wanted it. And they wanted it because they knew that if there were regulations, it would provide barriers to entry for smaller competitors. You know, they wanted the hurdles to be just high enough that you had to have a lot of money to be able to get a product on the market.
I see. So some gatekeeping.
Yeah. I don't actually buy that argument, but that's his argument.
Dr. Quinn, I have a question on when speaking about de-extinction. How does the fact that a large majority of species have been killed off due to anthropogenic, reasons affect basically bringing a species back, essentially?
I mean, it's the reason that people are even interested in it, for the most part, right? I mean, other than Jurassic Park, let's have a really cool zoo, which is not where most of the effort in de-extinction is. Most of the effort is in intervening to bring back, it's not just any species either, but so it talks about the Tasmanian tiger because it has particular import to the people of Tasmania. But as well, a lot of the efforts are to bring back things which serve important ecosystem services. And this actually, I was going to mention about the buffalo thing. There's actually some decent evidence that 10 to 15,000 years ago, a whole bunch of really big megafauna went extinct. And there's decent evidence that caused large-scale climate disruptions around the same time as the end of what we think is the last ice age, although it might not be. So yeah, I mean, it turns out if you take away hundreds of millions of large ecosystem drivers, and the situation on the ground becomes radically different. Now, there you get a problem with scale, because a lot of people, my students, like 98% of them will say, well, it's been 10,000 years, the situation has moved on. Even if it was humans that caused those extinctions, which is, that also is up for argument. And so the easy cases are the ones where it's something reasonably important to the ecosystem, something that's important to people, something that went extinct recently because of humans, and something that's not dangerous. So I've got friends who want to bring back wolves and grizzly bears and apex predators because a lot of us have never had the experience of being actually in danger of being eaten. And a friend of mine thinks that's actually an important human experience. So 99.9% of the... We actually got a letter from somebody who said that it's scientists like him that caused 9/11. Like, I don't know how you make that argument, but the point is...
It's kind of like blinded children.
Yeah. It's extremely... It riles people up quite a lot. There's a lot of... But they're good candidates.
We've got time for one.
Giant ground sloth. They can run away.
Oh, yeah.
Samantha, we've got time for one more question.
The woman in the back.
This is for Dr. Quinn. So, with the process of de-extinction, I understand that they're able to bring an animal alive, but how close are they to actually bring a species to be able to independently survive on their own?
Yeah, right. So first of all, that organism didn't live very long. So that's one problem already. Nextly, there are ways you can try to get a reasonable amount of genetic diversity, which is something I didn't talk about, but should have. This is another use of these technologies, is when you have a species like cheetahs that went through a genetic bottleneck, you might want to use these technologies to increase the genetic diversity to help the species out. So if you're trying to de-extinct a species, you need not just one individual, not just several, but you want a pretty good amount of genetic diversity. But probably I would say the main problem is where you're going to put it, because there's not a lot of habitats left. So you need people who are willing to have the thing be de-extincted and have a potentially large number of large animals running around that weren't there 100 years ago or whatever. So some of the movement in favor of de-extinction is coming from billionaires. Ted Turner apparently wants de-extincted animals on his ranch. He owns a whole bunch of land out there in Texas, and he's been talking to these people, which I think to me raises an important point that, like, for some efforts like this, there's a lot of counter arguments. You're taking money away from other conservation efforts. You know, every dollar you spend on de-extinction, you could spend in trying to protect the rainforest. But this example of Ted Turner shows this could potentially open up more funding that wouldn't otherwise be there and more interest that wouldn't be there. How much do you think, there's people who go to Africa to shoot elephants and stuff, and they pay like $300,000 to shoot an elephant, and then that $300,000 that gets used for conservation of elephants. Okay? And so there's some of that going on. How much do you think somebody would pay to shoot the first mastodon? That's been killed by a human in 4,000 years, say. That could be a huge pot of money for conservation.
Yeah, the other thing I was going to mention after your talk, though, is that there actually starting to be a lot of interest in using these technologies to help species that are threatened by climate change make adaptations.
Yep. Yeah, so one of the proposals is to bring elephants to North America, because actually Asian elephants are more closely related to mastodons than they were to African elephants. So bring Asian elephants here, put some genes in them so they can tolerate the cold.
Yeah. De-extinction of a species requires an ability to reproduce. Because if you really boil it down, every species has to reproduce to maintain its population. So if we want to think about de-extinction of some species, like take a mastodon or a woolly mammoth, how are you going to get those individuals to then reproduce if you can't make males and females that have some genetic diversity that can go on after that? So I think taking into consideration the reproductive capacity, should they be a single individual be generated?
And then behaviors.
Yeah, behaviors is critical. But I don't think we always think about this, right? Even if you want to make that one individual, if you want to bring back a mammoth, what uterus are you going to put that embryo into? Because I'm not sure of any species that has a uterus that size that can carry that thing to term, even elephants. So that'd be a logistical impossibility.
Okay, I hate to cut off the conversation when it comes to mastodon hunting, but our time is up. I neglected at the beginning of our event this evening to thank our co-sponsor, which is the School of Politics, Philosophy, and Public Affairs. This is an annual symposia series that we do with the school that looks at science, ethics, and public policy issues. I also want to thank my colleagues Samantha and Bill Cavasenci for helping us organize this panel tonight. It's been a fascinating discussion. Join me now. Oh, one other thing. I just want to remind you, tomorrow at noon we have the chair of the National Endowment for Humanities here. He'll be talking about a cultural policy for the 21st century. It should be a fascinating discussion. I encourage you to come to that. Join me now in thanking our guests for really a fascinating conversation.